So today I'm going to talk about my group's work in modeling biological cells, and in particular a vision to operationalize synthetic cells into global biotechnology. And what I'm going to build toward is a vision for a U.S. Institute of Cell Design as a research institute here at MU. So biotechnology is definitely part of our future, and quantum computing and AI are definitely going to get us there faster. And so I'm going to explain what I mean by that in 4 vignettes. The first one is that biology must become designable, not just explainable or predictable, but designable. And synthetic cells are the predicted chassis for a lot of this future biotechnology. And in fact, according to McKinsey consultants, upwards of 60% of the physical inputs to the global economy are going to come from biology: fuel, food, medicine, materials, et cetera, replacing petrochemicals and fossil fuels. In the second vignette, I'll talk about what I mean about cell design and new to nature functions, and the fact that these require not just biology and chemistry anymore, but physics, math, and compute. The third is, well, the physics is the Zia Labs brand, and I'll talk with you about what we mean by that. And then I'll talk about some discovery highlights from my lab and what's coming next. So all of this building, again, toward an institute for cell design. So synthetic biology, that's what I'm going to talk about a lot today, and it already permeates our society. It includes yeasts that make our food, E. coli making insulin. We do genetic engineering to reduce species loss or engineer crops, and we also engineer virus resistance, natural fabrics, as well as controlling algal blooms and pandemic agents. And so this is the power of what the world currently uses to sort of wrangle biology into doing things that we want it to do. That's how we use natural wild type biology. Now there are people really beating the drum that there's a profound transformation underway in the way that we make pretty much everything. And the idea is that we can execute some sort of transition away from fossil fuels to save the world. And it's actually a pretty comprehensive vision. And the idea here is that this would be this revolutionary potential would routinize biology as a general purpose technology. And that means deploying it everywhere, using the biology that surrounds wherever you are to make everything you need. That's the sort of grand idea. But the idea also that's stopping that is that the world is hitting a hard stop with what we can do to make nature give us what we want it to do. Natural cells are constrained by evolution, billions of years of evolution, and we've maxed out redirecting that at scale. So synthetic biology is essential to scientific leadership, and a lot of us are thinking, especially on the top row up there, about the Genesis mission right now and its main goal to do this. And we need it for things like drugs, biosecurity, biowarfare, and general global competition. And it's going to be needed for everything, virtually all drug manufacturing, food, fuels, emergent viruses or biological attacks, as well as sequestering critical minerals and encoding cryptography. All of this is going to be routinized through biology in the coming decades. So one takeaway that I want you to have going forward is that biology, and specifically cells, must become designable. Well, what do we mean by that? Again, I want to emphasize we cannot squeeze any more control from wild type cells, so we have to make new ones, synthetic cells. These are the factories of the future. And for them to be effective and controllable, they need to be a lot simpler than natural cells. And so where are we at in this process? What progress are we making for these little factories? Well, again, the takeaway is that definitional for synthetic cells is they have to be simple, small genomes, fewer biomolecules. And it's taken 30 years to get where we are today, starting from 1995, and the discovery and isolation of the smallest wildly occurring genome. And that was in Mycoplasma genitalium. And after the development of gene disruption technology and random transposon mutagenesis, these were stripped back to about 350 genes essential for life. And then this was followed by a development of genome synthesis and installation, and I talk about that casually, but this was like a tour de force in biotechnology and genetic engineering that brought from the J. Craig Venter Institute the JCVI they called SYN 1.0, and this was the first living, dividing, top-down synthetic cell which had about 500 genes, still not the minimal genome. And then in 2016, and this is the cell that we model, JCVI SYN 3A, which is the minimal cell capable of life. It's synthetic, and it has the smallest genome capable of the central dogma of living. It's still delicate, however, and it has no new to nature functions. It still does what nature wants it to do. And so where we're at now is a global descriptive modeling approach, but cell design is still out of reach. So genetic engineering and top-down synthetic biology gave us Syn3A. That's the takeaway here. It grows, it divides, it lives, but it's delicate, and it doesn't do what we want. It does what evolution wants. So to make biology, and specifically cells, designable, there are two musts. The first one is that we have to minimize complexity. And this means a bottom-up approach in building cells. And so this is an image from my collaborator, Kate Adamala, who actually builds the physical cells. I'm a computationalist. I have no wet lab. I don't actually make cells. She makes them. And the idea here is that they have layers of complexity that have evolved. And these create almost insurmountable barriers to making them do what we want. And the takeaway is that we're never going to get anything new from natural wild-type cells. The capacity that we need, in addition to synthetic techniques, is nearly atomistic control over all structure-property relationships from atoms to whole cells. Okay, so what that means is we need bespoke genomes and to make bespoke proteins in bespoke cells. And again, this is a cartoon rendering from my colleague Kate Adamala. And so predictive modeling, this is a second must, is what we need to give us what we want cells to do. Well, current approaches to doing that are Edisonian, cook and look. And there's just like millions of assays repeated over and over and over again. And finally you get that one, it's like the person looking for the golden ticket in the Willy Wonka factory, right? It's wildly expensive and very time consuming, as several have already spoken about today. What can we do? We can just flatten this thing. We can go all the way around this using AI tools and computational modeling. And I'm going to tell you a little bit about that and the novel tools that my group has developed to do this. So engineering can drive synthetic biology into a true design state beyond a descriptive science. And there are some approaches to cell modeling, but not all of them are design modeling. So these three main approaches, one that's very well known, is whole cell modeling using just kinetics. So there's actually no physics in these cells. They're flattened into metabolic pathway diagrams. They're extremely powerful and have given us immense predictability of functions that we already know about in cells. At the other end of the spectrum is people are attempting to represent every single atom and molecule within a cell. Just put the whole thing in there. And it's beautiful and it's been done, but the thing is that they turn it on and it goes, and then it sits there stuck, compute, compute, compute. And it would take like a lifetime of the universe for it to get through one cell cycle and all of the world's compute power right now combined. In between that is my lab's research. And so we do coarse-grained whole cell models. And our strengths are we can do long-time whole cell dynamics timescales. These are actual physical objects. You see a real nucleoid in there. And our weakness is that our biochemistry has still been limited. It's pretty good, but it's not atomistic. And so that's what my lab has gotten a lot of funding from DARPA and Sloan Foundation to now bring atomistic resolution into a whole cell model. And the spoiler alert is we're not going to get there with just physics-based models. We're going to actually need advanced AI and neural network techniques. So why is it so hard to get physical resolution and atomistic chemistry? Well, this is a really beautiful movie. It's a cartoon from Harvard BioVisions. I encourage you to look at this movie on YouTube, The Inner Life of a Cell. And it has music and it's super inspiring, and I still watch it when I'm like, completely like, oh, we're never going to solve this problem in research. I go back and watch this movie. But it's a cartoon. There's no physics here. Because doing these kind of physics over whole cell timescales, again, lifetimes of the universe. It's really difficult to do all the atomistic interactions for billions of atoms in a cell. Well, this is what we're doing. Using our innovative AI-enhanced pipeline, we are going to bring near atomistic resolution into our models. So we don't want to start with the whole thing. We want to pick one really key process in cells, and we've already gotten some good, strong success with that. So the first big swing should be something in the central dogma, something that everybody cares about. Protein synthesis. It's got to happen in a cell. 50 to 70% of the energy or effort in a cell is devoted to making all the proteins in all of your cells. It's happening in you right now. And it's done inside the ribosome, which you see here. And there's a polypeptide chain coming out at you. It's going to break off and form itself into a protein and do all the stuff that you need. And this is not our movie. This is one put together by the Nobel Prize winners who characterized what goes on inside a ribosome. But my group was the first to actually build a model of protein synthesis and to show how E. coli in particular speeds up protein synthesis when it needs to grow faster. And what we found and what we showed is that physics, not just chemistry, is essential to this. And this was a big paradigm shift in the community. So the idea, we were able to recover with our simulations predictions for in-cell, in vivo experiments, which we were really pleased with. And just even our early simple models explained this, that colloid physics really control protein synthesis rate. So that's the big takeaway is physics matter. And the other part is, great, we showed what a ribosome could do, but ribosomes don't live forever. Every cell in your body, its ribosomes are getting old, and then they're going to die, and then a new one's going to get made, and that's going to continue. It's the way that it goes. Growing and dividing and replicating cells have to make their own new ribosomes. And this turns out to be the sticking point in synthetic biology, ribogenesis. It's actually one of the biggest existential barriers to synthetic biology and synthetic cells. It's hard to do. Making them de novo actually has not been done yet. There's some really great in vitro methods like ISAT that are almost there, but they still have to hand feed it all of the ribosomal proteins. So it's not autonomous. I can't make a whole bunch of these cells and say, go forth and make yourselves and continue making all these drugs that you're making for me. I have to hand feed them, which again, wildly expensive. So why is it hard? And this is really important, this last key point, physical context is implicated. Remember, synthetic cells need to be much simpler than wild type cells to work for us to get what we need out of them. And when people make bottom-up synthetic cells, it's like they maybe threw away the baby with the bathwater. What's recognized by my colleagues that actually make synthetic cells is they've lost some spatial context and they don't know which piece it is. And they're back to the Edisonian searching for what do we need to bring back in. And so that's why my lab was reached out to ask how can we do ribogenesis and make that model. And that's what everybody in my lab is devoted to right now, using our recent awards from the Sloan Foundation and DARPA to model this process. So these are I like showing these images here. The top one was built by a lab member, Vishal Shivashankar, who's in my lab, and the atomistic simulations being carried out by Harnoor Sachar, who's also in my lab, and then the bottom one by Jesse Rory as well. And what do? Our aims are to understand and control from the Sloan Foundation grant, how does matter actually become life? So we're asking that question and guiding development of synthetic cells. That's the other piece that we're doing. So we have these two axes of inquiry. Our method is to get near atomistic resolution into a whole cell model and eventually marry this with quantum computing. And our strategy is to train neural networks to make the whole cell model go fast. We use atomistic simulations to generate ground truth data and then generalize it. So this is ribogenesis, a super idealized version here. It's actually in a eukaryote. So there's DNA and it's being transcribed, and that yellow thing coming out is supposed to be a ribosomal RNA. And it's just so beautiful because it looks like the first part of the ribosome is being synthesized, and it's all so orderly. And I call this a ballet idealization because in reality it's ridiculous. It looks like it looks like Penn Station. I've been in Penn Station, or an airport at the peak maximum time where everybody's running around like crazy. It's a wildly complicated place, and nothing happens deterministically like this other than the transcription. So it's a complex multi-step process in a crowded, highly diffusive cytoplasm. And for it to be modeled, you have to get the atomistic structure to do molecular dynamic simulations. And it's hard enough to do that with two molecules. Am I right, Jack? In an MD simulation, it is hard enough to do that. You suffer greatly to do that with two fairly small molecules in a simulation. But we've got 54 of them, and they're big and long and complicated. Again, you just go home crying if you try to do this with physics-based modeling. Just can't get there. So we have developed an AI-guided multi-scale modeling framework to design this autonomous self-assembly in the 30S subunit. And I'll just give you a quick snapshot of how we do that. We know here with a representative protein that about 18 atoms make up one residue. Those are the little bits that do all the interacting and the binding that was discussed earlier. And then we use something called Martini. It's a wonderful force field algorithm that is widely recognized and being used for all sorts of things in drug discovery to turn every about 4 or 5 of those into one highly functionalized and intelligent bead. It still carries all the atomistic information, and then you've just got a lower computational burden, and they have been proven over and over and over again in drug discovery and design. We're taking this one step further. In the model system that we're designing in my lab, we have developed a neural network model approach where we now turn those 4 into one highly intelligent biomolecule. And so now we've got this coarse-grained mesoscale thing that I can actually do in a whole cell. And we can go from that atomistic information and retain that all the way at the whole scale level. So that's what they're paying us to do right now. It's a cartoon of how complicated it is. Here's a snapshot of one of Harnor's simulations where he's showing, yep, we know how to do this. And I also wanted to give a shout out, also all of this possible because of our colleagues in Research Support Services, Matthew Keeler and Jennifer Nixon and all of the staff over there and Hellbender who helped us spec out and identify our rip-roaring fast GPUs that we just installed. So overall, our AI-driven physics-based modeling is leading the way forward, and our ground truth simulations will be used to train our neural networks. And we don't need huge, massive tranches of data to do this because it's just amino acids and nucleotides, and our model can generalize. And we're in the process of putting in a proposal for the Genesis mission to multiply this effort, and our general vision is go from phase 1 to phase 2 to a center here at MU. And yeah, so where? Here I had AI. I'm like, I know this was the old building, but I'd like that floor. Please, Prasad, thank you. All right, good. And policy is key. I was just at a biotechnology leadership summit yesterday, and we're in the billions for the Large Hadron Collider and the James Webb Telescope, and we're in the trillions for data centers, and we're still only in the hundreds of millions for investment in biotechnology. So we have a lot of progress to do there, and I just want to acknowledge my former lab at Stanford, my current lab here at MU, and also ERA, who is keeping all of the wheels running. They are the most, can I have a shout out for ERA? And with that, I'll take any questions.